next thing we want to talk about is nerve impulses. The neuron membrane is designed and specialized for communication. The messages that the nerves send to one another are called nerve impulses or action potentials. Basically, this is going to be an electrochemical signal that's caused by sodium ions and potassium ions moving or passing across the plasma membrane through ion channels. So what are ions? Does anybody remember from chapter 2 what an ion is? Ions are going to be charged atoms. They've either lost or gained an electron. Ions are able to pass through the plasma membrane on their own. The question is, are ions able to pass through the plasma membrane on their own? Does anybody remember when we talked about what types of molecules can pass through the plasma membrane? Not ions. Any molecule that's too large or too charged won't be able to pass through the plasma membrane. Ions are going to be too charged. They carry a charge. They are not able to pass through the plasma membrane. So they're going to have to go through what are known as ion channels. So what we're going to do is going to pass through these ion channels. Each ion channel is going to allow only certain ions to pass through it. Ion channels might be permanently open, or they could be regulated by a gate, which is just a protein that changes shape and either opens or closes the channel depending on the particular needs of that cell. The transport does not require any energy, as the ions are going to follow down a concentration gradient. This is going to be through, they're going to follow that concentration gradient, moving from areas of high concentration to low concentration. Does anybody remember, is this what's known as passive or active transport? It's going to be our passive transport.
Cells in the nervous system are the neurons. In addition to the basic cellular elements, neurons have a variable number of slender branched extensions called dendrites, which receive signals and direct them to the cell body. A single axon carries signals away from the cell body towards synaptic terminals, the sites where neurons communicate with other cells. Signal propagation within the neuron begins where the axon exits from the cell body. All of the action in this first step takes place on either side of the thin plasma membrane that constitutes the outer border of any animal cell. The cellular components are found inside the plasma membrane. Outside is the extracellular fluid. The fluid inside and outside a cell contains many ions and charged molecules. The difference in the concentrations of ions inside and outside a cell results in a concentration gradient across the plasma membrane. Sodium ions are more concentrated outside a cell than inside. Potassium ions and negatively charged protein molecules, abbreviated A-, are more concentrated inside a cell than outside. The distribution of ions and proteins creates a separation of charge across the plasma membrane. This separation of charge represents a form of stored energy called a membrane potential. If we use a voltmeter to measure this potential, we find that the interior of the cell is negatively charged relative to the exterior. Ions move across the plasma membrane through protein channels. Most channels that permit the movement of charged ions across the membrane are gated and remain closed in a resting neuron. These channels rebuff sodium and potassium ions that would otherwise move through the membrane. However, one type of potassium channel, sometimes called a leak channel, is always open, allowing potassium ions to cross the membrane. Because the inside of the cell has a greater concentration of potassium ions than the outside, potassium ions will tend to diffuse down their concentration gradient to the outside of the cell. However, the positively charged potassium ions are also attracted to the negatively charged cell interior. In a neuron at rest, these two forces, diffusional and electrical, are in balance, and there will be no net movement of potassium ions across the membrane. For every potassium ion that leaves the cell, another returns. The voltage-gated sodium and potassium channels open in response to specific changes in voltage, which may occur when the neuron is stimulated by another cell. When an excitatory signal reaches the axon, the voltage-gated sodium channels begin to open, and a few positively charged sodium ions enter the cell, going down their concentration gradient. This causes the inside of the cell in that region to become less negative. If the signal is strong enough, a chain reaction is set in motion, in which more and more voltage-gated sodium channels open and sodium ions rush into the cell. The influx of positive charges results in a rapid reversal of polarity, or relative charge, across the axonal membrane, and the cell interior at this location now becomes positive relative to the exterior. As this happens, the sodium channels snap shut and the voltage-gated potassium channels open. Potassium ions rush out of the cell, and the charge across the membrane returns to its resting state. Meanwhile, the changes in the membrane potential have caused adjacent voltage-gated sodium channels to open, permitting sodium ions to rush into the cells and causing a rapid reversal of polarity across the membrane at this new location. This voltage change causes voltage-gated potassium channels to open, and potassium rushes out of the cell, returning the membrane potential to its resting state. In this way, the action potential is propagated along the length of the axon. You have now seen how the membrane potential depends on the relative ion concentrations on either side of the plasma membrane. These concentration differences are maintained by an energy-consuming pump in the membrane called the sodium-potassium pump. If this pump did not operate, the differences in ion concentrations across the membrane would soon disappear. With each cycle, the pump takes up sodium ions from inside the cell. ATP phosphorylates the pump, giving it the energy to change shape and expel the sodium ions to the outside. The pump then picks up potassium ions from outside the cell, releases its phosphate group, and returns to its original conformation, releasing the potassium ions inside the cell.